Hello again, everybody. Thank you for joining us today. My name is Cristel Rabil, and I am the Genesys assistant at the Crop Trust. And welcome to today's webinar called From Bean to Browser, Genesys for Coffee Collections. This webinar is a follow-up to a workshop that took place in Ivory Coast in May that was organized by the Crop Trust. And it was for access and benefit sharing for coffee collections. So thank you for those who joined that workshop and who are joining this follow-up webinar today. And also thank you for all our partner gene banks who joined us today, because we opened up this webinar for more gene banks to join. I will start this webinar just by giving a quick, brief presentation on what is Genesys for those of you who are not familiar with the platform. So Genesys is an online platform where you can find information on more than 4.3 million accessions in crop gene banks worldwide, covering all crops for food and agriculture. Genesys development and operations are currently funded by the Crop Trust Endowment Fund. Genesys focuses on accession level data about material in long-term ex situ conservation. So Genesys does not include material that is strictly in situ unless it is formally part also of an ex situ collection. And also Genesys does not host information on breeding materials or released varieties unless they are part of a formal gene bank ex situ collection again. So at the core of Genesys data is passport data. And you see on the right-hand side a snapshot of a table of passport data example from Genesys. This is the rice conserved at Africa Rice Center. Briefly, what this passport data includes, it includes information on where are the accessions currently conserved, the accession numbers and names that are given by the host holding institute, and also information on collecting institutes and collecting codes and missions, as well as information on breeding institutes, if that material was bred and then conserved in the gene bank, on safety duplication institutes, if this material is safety duplicated in Svalbard, for example. Information on, of course, the taxonomy, so the scientific names and authorities. Information on collecting sites, so we include geographic coordinates as part of the passport data, and among other information. So currently, Genesys holds, again, information on more than 4 million accessions coming from over 500 different gene banks. And those partner gene banks are international, regional, and Article 15 collections, such as the ones that are highlighted in pins. So all the green pins are the international CGIR centers that you see. And the red pins are the Article 15 collections. We also have the regional gene banks in orange, for example, the Southern African Regional Gene Bank Network. And in green, for example, the Embrapa network in Brazil. In brown here is the USDA networks of gene banks in the US. And in blue, the Eurisco ACPGR network of gene banks in Europe. But we also have data from national partners, and those are highlighted in red, that we are currently focusing on increasing the number and partnering more often with. So I already mentioned the passport data in Genesys, what it includes, and this passport data is all standardized, so it's coming from more than 500 gene banks to us in different formats, that we make sure that this is in Genesys all following then the format of the multi-crop passport data descriptors in its latest version, version 2.1, and this covers all aspects of passport data, such as identity of the gene bank, identity of the accession at the gene bank, the taxonomy, status in the multilateral system, etc. And beyond passport data, which is the core of information in Genesis, we also include accession images and scanned collecting forms that can be attached to individual accessions. Also, gene banks can upload collection subsets such as core and mini-core collections, reference sets, etc. Also, gene banks can upload their own descriptor lists that they use to characterize their collection, as well as trade data sets, which is what we call evaluation, characterization and evaluation data sets, which we also have from many different gene banks in Genesis for different crops. I share here with you examples of how the images are visualized and displayed in Genesis. This is an example of a subset. It's actually a mini core collection. Now I will move on to share with you the benefits of having your passport data of coffee in Genesis. So once your data is in Genesis, a suite of tools becomes available to you as a gene bank, as well as to the users, like researchers and breeders that are interested in your gene bank coffee material. For example, we have trade data querying directly on the platform. So right now, Genesis allows you or your users to filter for accessions based on their traits. For example, you can filter for the coffee accessions based on their caffeine content. If you have this uploaded in Genesis, then you don't have to download separate files and filter those in Excel or separately. We also have the option to query passport data using an AI search, which we recently added on the platform. It's you can prompt Genesis using natural language without having to know how to use every filter of passport data in the platform. You can just write sentences and prompt this Genesis AI, and it will return to you accessions that conform to your search. We also have a way for you to narrow down the accessions based on the climate at their collecting site. So in Genesis, if you upload accessions with geographic coordinates of where they have been collected from, Genesis then matches the accessions with the climate profiles at their origin using a separate data set, the WorldClim data set in its latest version. And you can then narrow down the accessions based on the climatic indicators that you choose and narrow down these accessions. We also have created a tool in Genesis in collaboration with SIAD and ICARDA to allow you to create subsets based on agroclimatic variables. So think of it as a DIY, do-it-yourself subsetting directly on the platform based to create groups of accessions of coffee, for example, that have similar, for example, maximum temperatures at their collecting sites. We also have the option to find similar accessions held in the different gene banks or even within your own gene bank. Once the data is published in Genesis, you can run the similarity search tool, and this allows you to find duplicates within your own collection or duplicates between your collection and other collections and other gene banks that also publish in Genesis. As I mentioned before, also you have the option to upload images and documents and link them to your accession passport data. Genesis also then generates different types of visualizations of your data, such as in overviews and charts, and heat maps or geographic maps, with the option to overlay on these geographic maps other maps like climate maps, for example, of maximum temperature or precipitation. And then you can download those visualizations from Genesis and use them in publications, or if you're needing those for presentations. Currently, we have Genesys. All of the platform is translated to French, Spanish, and Brazilian Portuguese. And we're currently working on adding more translations on the way. So you can also access this data that you have in your gene bank in different languages, and therefore your breeders and researchers who are interested in your material can also access your data in different languages. If you are a gene bank data provider to Genesys, then our team can send you on demand a report on passport data quality and completeness that includes tips on how you can improve the completeness and accuracy of the data about your coffee collection. This focuses on passport data, and it's a very extensive about 20 or more page report that focuses on all the aspects of your data, such as the taxonomy validation, the geographic coordinates validation, and I will show you an example in a bit. Also, the platform allows you to receive material requests from users that Genesys relays to you to your gene bank, and then you can take it from there with the users separately. And finally, Genesys includes a tool that allows you to mirror all of the data and all of the tools that I already mentioned that are in Genesys, such as the material requests, the translations, the similarity search tool, or the trade data. It allows you to mirror all of these tools and your data that is already in Genesys on your institutional website, if your gene bank has its own website. And this would be synchronized with Genesys. So whenever you update data in Genesys, it automatically reflects on your own website. And with this solution, you don't need to maintain a separate database and update it in parallel to updating other global platforms such as Genesys. So this would be a time-saving tool for you that allows you also to have the data and the tools on your own website. I will zoom in here on the report on data completeness and quality that our team generates. So this is an example of one part of the report that focuses on the validation of geographic data. It gives you some clues on how many accessions need corrections, how many accessions are falling in the ocean based on our validation, how many have mismatched countries, mismatching their latitude and longitudes. It narrows down these accessions then and gives you like tips on what you could fix in this data in order for example, the country to match the coordinates, or in order for the accession not to fall in the ocean. Of course, these are only tips, and you, at the end of the day, make the judgment on how it's best to fix those accessions. But at the very least, you can use this report as an indicator of which accessions are flagged that need to have something corrected in their data. This is only a small snapshot of the geographic validation in the report, but it also includes parts on missing taxonomic information or improvement on misspellings or inconsistencies in the taxonomy. Same for the geographic data, and some gaps in key management data, for example, storage type or biological status of the accessions that are missing that you can add, and provides tailored suggestions for you. Even if you're not currently providing data to Genesys and you want to use some validation tools, so we put out there the validator tool it's publicly accessible right now, and it validates the taxonomy and the geographic information, but it doesn't generate a report with tips. It only gives you results on what are the accessions that have, for example, misspellings in their taxonomy and suggested fixes, but it's not like in a comprehensive report as the one I showed here in the slide. It shows you direct results line by line, and then you can, of course, implement the fixes. This is a web-based tool. It's a website, and I share with you the link here. And it's open to anyone and does not store any data, of course. I will share the link in the chat. This is how the tool looks like on the website. You drop your data in this first step, and then you choose at the very last step between either validating the taxonomic data, the country of origin, or pinpointing if the accessions are falling on the land or water. And there we have a YouTube video, brief demonstration on how you can use this tool. So I will share the link in the chat right now. Right, so back to our presentation. I want to now present to you how you can submit and update passport data in Genesys. Maybe a lot of you I see here already have are partners with the Genesys and have signed the data provider agreement. But for those of you who haven't, the first step in order to be able to share data in Genesys is to sign the data provider agreement with the Crop Trust. This is an agreement that allows us as Crop Trust and Genesys to publish data on behalf of your genebank while giving your genebank full authority and full basically credits that this is your data, it remains yours, and you can delete it whenever you want and manage it however you would like. The second step would be, after signing this agreement, your genebank staff can then use the Genesys uploader tool, which is a tool that is downloadable locally and available at this link with its tutorial and user guide. And then after downloading the uploader tool, your genebank staff can upload passport data first in the test environment that we have just for testing purposes, and then later on in the Genesys official website. Just because we get this question a lot, Genesys, with all of its tools and the support that is provided by our team, is 100% free for genebanks. This is all coming from support and funding from the donors of the Crop Trust Endowment Fund. As the agreement is non-financial and it only is about data sharing and rights. I want to share with you now next a case study on improving alfalfa drought tolerance for Kazakhstan and Kyrgyzstan. This was a case study that was done by our partners in Australia, in the Australian Pastures Gene Bank, that worked on a project with the Crop Trust with partners in Kazakhstan and Kyrgyzstan, and we're able to use the Genesys platform, one of the tools that I mentioned to you, which is the climate search, in order to narrow down alfalfa accessions that have drought tolerance, so in Kazakhstan and Kyrgyzstan. So very briefly, I will share with you first of all, the problem that they were facing, the problem of declining water resources in the Kazakhstan and Kyrgyzstan region, which affects alfalfa growing conditions because alfalfa needs a lot of water to grow. And it is at the same time a very important source for farmers, for human use, and also for animal feed. So it has multiple end uses for dairy production, for beef production, lamb and goat, eggs and poultry, fiber, hay, seed, and honey production. So, the very strong point of the alfalfa is that it is a local commodity that is not subject to international price fluctuations, which is why it's very important for the communities in Kazakhstan and Kyrgyzstan, as we've seen how important this is after the war in Ukraine. So the problem again, it was that there was recently droughts that were affecting the region, so no water for irrigation, mainly in the summer, and that the local varieties have been slow to emerge from winter. So there were winter dormant varieties slow to emerge from winter. And the opportunity there was to develop more winter active alfalfa cultivars that have faster growth rates in spring and autumn. So in order to maximize growth when soil moisture is available from local snow melt and rainfall. And at the same time, plants need to be resilient to summer drought. This is the most important aspect for irrigation, because the water would no longer be available when the temperature reached 37 plus degrees. But at the same time, the plants need to survive very low temperatures during winter, minus 20. So I will focus only on the aspect of finding the drought tolerance. So out of the Metigago Sativa accessions that were available in Genesys, that have over 3,000 accessions that have geographic coordinates, the team from our Australian partners were able to narrow down these accessions to 26 from 3,000 plus. And the indicators that they used in the climate search tool in Genesys were 8 different ones pertaining to northern latitude, elevation, but also annual precipitation, annual temperature range, maximum temperature of the warmest month, and the mean temperature of the warmest quarter, among other indicators. So we have now the subset of 26 alfalfa accessions that have the similar drought tolerating traits based on these agroclimatic variables. The subset is currently available in Genesys, and I use this as a case study of alfalfa, but it could be applied for coffee or any other crop. The important thing in order to be able to apply this tool is to have passport data in Genesys. That includes geographic coordinates of the collecting sites, which later Genesys can then mash up with external climatic data sets such as the World Clim in order to narrow down these accessions based on this climate. I include to you here some general resources that I will be sharing with you as part of the presentation. After this webinar, we have general resources on how to use Genesys that could be relevant to you, or if you have breeding or research teams within your institutes. I won't spend a lot of time today on demonstrating how to use the tools. I just listed them for you to show you the potential. But we have here previously done a lot of webinars on how to use Genesys, how to filter passport data using the different tools, but also how to explore, how to explore subsets and trade data. And we also always share recent news in the Genesys blog post, on Twitter, and in the newsletter. I included the links for you. And you can always reach us on help desk at genesys-pgr.org. For the gene banks that want to provide data to Genesys, whether you already have signed the data provider agreement or not, if you want to provide data, we have some documentation on the basics of passport data documentation and the MCPD standard. We have some manuals that help you prepare your passport data, such as doing the data cleaning, mapping it to MCPD standard, doing the validation using the validator tool. We have this in writing, but also in video. And we also have a tutorial on how you can download and start using the uploader tool to start publishing data, either in the test environment or in Genesys directly. And we also have some manuals on how you can prepare and upload characterization data in written tutorials and video playlists. And if you are interested in embedding the data that is later in Genesys, if you want to embed it on your own institutional website, we also have articles and videos that show you how you can do that. I will, before moving on, I will share with you a few examples of those little tools like embedded Genesys and the trade data that we have. So just to explore more directly on the Genesys website. So we already have a partner that shared a coffee data with us. I will focus on CNRA in Ivory Coast. Just to show you how, if you have coffee data in Genesys, how that would look like. So CNRA's landing page in Genesys is here, and they shared with us around 8,000 accessions of coffee, of different species of coffee. And Genesys then shows you these charts and overviews on how these coffee accessions are distributed across the different species names, across their provenance, so where they have been collected from, etc. I will click here on the accessions number, and this will redirect me to the stable view that also includes more information, for example, the biological status of the coffee or the geographic coordinates. In the overview tab, you can re-access those charts. And in the map tab over here, you can visualize the geographic distribution of those accessions from CNRA. So from 7,000, almost 8,000 accessions, 4,000 have geographic data. And you see here, they are distributed in Africa. So what you can do in this case is use this climate search where you can narrow down these 4,000 accessions based on different climatic variables that I will show you here. So on the left-hand side, we have all the Genesys filters, and at the very end is the climate at origin. So those are 19 different filters pertaining to either temperature or precipitation variables. So just for the sake of example, I will choose to narrow down these 4,000 accessions based on the annual mean temperature at their collecting site, and I will choose I will choose a minimum of, for example, 20 degrees Celsius. So I will enter 20 in the minimum side and apply filters. So you see here, I've narrowed it down to 3,000 accessions. I can also even choose to use another variable, for example, the maximum temperature of the warmest month. I want it maybe to be at most or at minimum, I want it to be 30 degrees. So I narrowed these accessions down further. And maybe I want the annual precipitation to be an average of minimum, let's say an average of maximum of 500 millimeters. So I narrowed down now these accessions to only 56 that match. And all of them show right here. So I can access them on this table if I switch to another tab, and I can download them, for example, an Excel file. Or what I can do even is to request them directly from CNRA by adding them all to my list. I just click here and add to my list. And you see the 56 are now in like a shopping cart, this list in Genesys that allows anyone to request the material directly from CNRA after narrowing it down using the climate search. I will go back to the map view and maybe remove a bit of those filters to show you then how you can also overlay this map with another map, for example, of annual mean temperature that you can then download by printing here. And you can even choose the opacity. So this is the transparency of the overlaid map. Of course, there's images and different tools, but because of the time constraint, I want to now jump and show you, other than passport data, I want to show you subsets and characterization and evaluation data sets. So in Genesys, you can also upload subsets of accessions that either are representative of the diversity of coffee in your collection. So you can upload core or mini core collections of coffee. Or I show you here an example of okra from the World Vegetable Center, a core collection that lists here around 160 different accessions in the subset that are every link points to their passport data in Genesys. Other than core collections, there might be some selective types of subsets. For example, here the foxtail accessions that are tolerant to salinity. So these are 9 accessions that have been identified by ICRISAT to have a high tolerance to salinity. These 9 accessions have been identified from their foxtail millet core collection. So users can then, users of Genesys, breeders or researchers can then order these accessions also by adding them to their list here. Now it's 65, I added those to my list, and they can order them directly from ICRISAT. So you can do the same for your coffee collection. And more importantly, maybe for breeders or researchers, are the characterization and evaluation data sets that show trait by trait the observations of your coffee collection. I will give you here an example. For example, of the World Vegetable Center characterization of lablab bean. So these are 204 different lablab accessions characterized over 52 different traits. By clicking here, this green ribbon, this will redirect me to the table view of these characterization data, where I can filter, so narrow down these 204 accessions. I can narrow them down by, for example, the accessions that have the vein color of fully developed primary leaves of purple. So I narrowed it down to 70 items, and maybe I want to then download this data or add all of these lablab accessions that have purple as the vein color of their fully developed primary leaves. I can add it to my list and request this material directly from World Vegetable Center. I think we already have a coffee data set for an example that I will show you here. We have 2. So we have a phenotypic characterization of coffee accessions from CNRA. So these are 2,000 accessions of coffee characterized across 7 different traits. And perhaps the most important one would be the caffeine content. So you can narrow down these 2,000 coffee accessions, for example, to the caffeine content, for example, 3 or more. And so those are 32. So this is 1% of the data set. Of the almost 1% of these accessions have the caffeine content of 3 or more that maybe you might want to highlight, or if any researchers ask for this in your gene bank, you can then redirect them and point them to these 32 accessions, for example. We also have descriptor lists of coffee. So those are published by Kache, but you can, of course, publish your own descriptors that you use to characterize your coffee if you create your own using your own units and your own versions. Those don't have to be only the internationally developed descriptor lists. And you can then link those descriptors that you create to the characterization and evaluation data that you then upload in Genesys. So we also have a lot of material on how to use these tools that I just showed you, the maps, the overlaying of the maps, using the climate search, exploring the trade data, how to request material. So we have the very detailed steps documented that I will also share with you to have a look at, and maybe share with your researchers, or even for you to explore what is already in Genesys if it's interesting for your gene banks. And also the way to create a Genesys account, because all of these tools that I showed you would need an account just for you to be able to access and download, for example, download the files. We just do this in order to prevent robots from overloading the platform. So you can create an account for and explore all of these tools and access translations and use all the tools that I showed you. So with that, I thank you very much for your attention and for you for joining.